It's U of L Today on 93.9 The Ville. Here's your host, Mark Hebert.
And welcome to UofL Today with Mark Hebert on 93.9 The Ville. The UofL researcher has essentially stumbled into a new local anesthetic that is not in the cocaine family. Jeff Petruska is an associate professor in the Department of Anatomical Sciences and Neurobiology, and he's here to talk about his discovery. Welcome, Jeff.
Thank you, Mark.
Good to see
nice
you.
To be here.
All right, I said you stumbled onto this thing.
I did.
Which you kind of did, didn't you?
Yes, I did.
We'll talk a little bit about, just start off and talk about what you've discovered and what problem you're trying to solve here. And then we'll kind of go into some background.
I wish it was a situation where I'd spent a career trying to design the next best thing, but I didn't. I just had my eyes open at the right time. We used a drug that's actually been out there for a long, long time in a new way, and it did a great job. Local anesthetics are drugs that block the electrical conduction from the periphery getting to the nerves. So they're used very often to allow surgical procedures and prevent pain. All the existing local anesthetics are derived from cocaine. And although that might sound like fun, it's actually really, they do a great job, but they can be very, very toxic in the right setting. So because our drug comes from a family that's not cocaine derived and it has a long clinical history, there's a really good chance that it's not going to have the toxicity issues. And so we've started dealing with some folks who are experts in needs where cocaine-derived local anesthetics can't be used, such as corneal pain, eye pain. The existing local anesthetics, if you put them on there for more than about a day or so, can start to literally degrade your cornea, so they can't be used. And so that's one of the places we're looking right now to see if we can use this new drug to offer some pain relief.
Okay. And this would be used for eye pain, correct? Mainly is what you're looking at. I didn't even know you could put something directly on your cornea, on your eye to stop the pain.
Very few people know about that unless you've been to the doctor. A lot of people go in and they'll get a very short-acting local anesthetic so the ophthalmologist or optometrist can do a test. But you can't use that drug for very long or it'll degrade your cornea. So when you get long-lasting eye pain, there's really not much out there for relief.
When you talk about long-lasting eye pain, that wouldn't be something like you get a stick in the eye and you got to get a little piece of stick removed or something. That's something that's more degrading to your eye, or what are we talking about?
Even that can last for many days. That would be a corneal abrasion or an object in the eye. So in those conditions, if you've got pain, sometimes it can be very severe for a few days. You still can't use the local anesthetics that are out there. Ours would be useful for that. Then there's conditions like dry eye. And if it's severe dry eye, people can have pain for a very, very long time. There's no cure for dry eye right now. Our drug might offer some relief in that condition. And so we're working it up right now with that in mind.
Okay. And I did say that, as we said off the top, that you kind of stumbled into this. So what were you looking for? What were you searching for? And then you kind of stumbled across this new potential drug.
I was working with a set of anesthesiologists who were trying to work with some drugs that could make the existing local anesthetics last longer. So there are a lot of conditions where we want to be able to use local anesthetics, but you can't keep adding them because of the toxicity issues. And so what they've started doing now is adding all other drugs that make the local anesthetics last longer. And in the process of those experiments, I ended up putting a drug onto a nerve. It hadn't really been put on nerves before, and it ended up doing this job all by itself quite well.
And you said, hey, this works. Look at this, guys.
I really thought I'd messed up what I put on the nerve.
But it worked okay.
Everything worked great.
Go figure, right?
Yeah.
Well, you're glad you stumbled into it, obviously. So
yes.
Where are you in the process of testing this new drug, this anesthetic, and how far along are you?
We are fairly far along in terms of the proof of principle. We did some experiments to convince ourselves that this was real. Very fortunately, the University of Louisville has an excellent technology development and commercialization program in place. A lot of folks around here have been doing it. There's a lot of experience. We have a new grant that's a partnership with the NIH called the Excite program, and I was awarded one of those grants. And it's a 4-stage grant. We're halfway through it right now. So far, everything has come out the way that it needed to advance this on to the next step. So we're very happy with this. And now we've actually started a company. We're going to license the technology from UofL, hopefully, and bring it out into a real thing.
How long are we talking about before it's brought out onto the market? Any idea?
Well, it's a long process.
I know.
It could be as quick as maybe 3 years. It could, of course, take anywhere 8 to 10 years. But because it's been out there for a long time, there's a really good chance we can use some of the prior data as backup. But it has to go through FDA, so it's going to take a while.
They got to have federal approval. Is this the first drug that you're aware of, first anesthetic that is not in the cocaine family that you can be used on your eye or eye pain? Well, or is there something else already out there on the market?
Experimentally, there are drugs that are not in the cocaine family that can do this very same job. The problem is, can they be used efficiently in the clinic and safely in the clinic? And generally the answer is no. There are some drugs that end up acting as local anesthetics, but it's very anecdotal. They're not approved for that purpose or anything like that. So the answer to your question is probably would be the first.
Okay. We're talking with Jeff Petruska, who's in the Department of Anatomical Sciences and Neurobiology at the University of Louisville, and he's working on a local anesthetic that hopefully will work and hopefully will go to market here in somewhere between 3 and 8 years. What did you learn through this process?
I learned a lot about how the system works on the other side. Basic science does one thing. We have all of our goals and principles in mind, but those do not always mesh very well with how you actually bring things into reality for clinical purposes. I also work in the Spinal Cord Injury Center, and one of our goals is to bring treatments out. And it's not always what we're taught as scientists, and it's a different set of rules and a whole new language I've been learning with this commercialization effort.
Did you also learn that you have to be able to look beyond what you're focused on?
Absolutely. The eye pain is wonderful. I'm actually a pain expert. It's one of the things I study. And I'm a little embarrassed to say that I didn't realize that there was such a need for corneal pain relief. But it's been out there. And so, exactly as you said, you have to get out of what you know, go talk to people that do things. Patrick Scott, a friend of mine from
We've had him on the show.
Exactly. Was the one who actually made me aware of this. And because we talk to people outside of our field, we learn these things, and it's incredibly important.
And you also said that University of Louisville has been supportive in terms of getting you grants, and you talked about was it Coulter grant that you got, or the
we have the Excite grant.
Excite grant. Have you found that the University of Louisville has had the infrastructure and the wherewithal to help you through this process?
I've been very impressed, actually, with the expertise, the advice, the willingness and openness. I think it's a very nice infrastructure that's there. It is not pervasive. You do have to seek it out, but once you identify it and find it, it's there, and it's really good. I'm very happy with it.
Okay, and before we go, what's the name of your company that you formed?
It's called Desense.
Desense.
As in desensitize.
Desensitize. Desensitize the eye. All right, very good. All right, Jeff, we appreciate you being on the program. Thanks for coming on. And when you get further along in this process, maybe 2 years from now, we'll have you back on and you can talk about where you are in the process on this one.
Pleasure. Thank you for the invitation.
Are Kentuckians engaged in their politics? How does our engagement in government compare to other folks in other states? Well, Glippy Grider-Jones is with McConnell Center, which has been a partner in the Civic Health Index for the state of Kentucky. Welcome, Glippy. Good to see you.
Thank you for having me again. Glad to be here and glad to help start this conversation about Kentuckians and their civic engagement and involvement. So,
all right, so I've got this study sitting in my hand right here. It says the 2016 Kentucky Civic Health Index. What is the Civic Health Index?
Well, the McConnell Center is partnered, and I definitely want to give credit to our partners as well. We're partnered with the National Conference on Citizenship, as well as Western Kentucky University, and then we've also collaborated with the Kentucky Secretary of State's office, so Secretary Allison Lonergan-Grimes and her office. But we are all partnered, and we've helped produce this report. It's the 2016 index, but it's a 5-year update on our inaugural report, which was launched in 2011. So in a lot of ways, and I think once we dive into a little bit of the data, it's interesting. Some people may say, well, it's only 5 years of data. How many elections have we had since then? How many metrics can you really engage? And that is a good point. There's maybe not some alarming information here, for instance, but it is an update, and it's a 5-year conversation point, and it's really helpful because I think it shows where we were 5 years ago, which was our baseline. We didn't have the data before that, but it shows now some movement. The question is, are these anomalies in the movement, or are these a larger trend? So obviously, hopefully in 5 years, we'll have 10 years worth of data and can speak more knowledgeably. But it's a great flashpoint. It's a great snapshot, I think, of some things we're doing really well in civic engagement, and obviously some things where we
We're
can
not doing it very well.
certainly improve, I would say.
So what is civic engagement? What do you know, in a broad spectrum, like you said, we'll talk about a few specific things here in a minute. But when we're talking about civic engagement, what does that mean? I go to vote.
Yeah, sure.
I call my congressman. What does it mean?
Yeah, so those are definitely some formal measures. Obviously, voting is probably the most popular electoral voting. Other formal elements would be things like boycotting, things like contacting your congressmen and women, sort of these more formal metrics. But then the report also has, and in particular, has some new metrics as well that really dive into sort of these informal measures, things like social connectedness. You know, how well do you really know your neighbor? How well are you really engaging in your community? Volunteerism is another key metric that the report highlights. And then the new measure that I hope we do get to dive in a little bit, confidence in public institutions. How much do we trust the media?
Well,
Go ahead and go there.
Since you went there, that's what I want to talk about first, because I thought I read the report and I thought that was the most interesting thing that was in the report. Again, we're talking with Lippy Grider-Jones from the McConnell Center at the University of Louisville about the Civic Health Index in the state of Kentucky for 2016. So confidence in public institutions. Why don't you talk a little bit about the media, first off?
Sure.
Let's start there and talk about when you polled Kentuckians, what did they tell you about their confidence in the media?
So this measure, and again, I want to be clear here, I didn't author the report, but just help launching and help starting this conversation here. But the key measure for confidence in the media in particular, there's a big gap between where Kentuckians are compared to the national average. There's a 10-point gap. That's one of the largest gaps between where Kentucky is compared to the national average. Kentucky ranks 48th. Let me say that again, 48th out of 51, because we included D.C. in the poll, but 48th out of 51 in trust in the confidence in media.
So what percentage of Kentuckians trust the media? Do you have that number?
I think offhand, I think it's like in the low 40s. Yeah, I would say I'm not eyeballing it right here, but low 40s. But I do know that women do have slightly higher, actually, I think it's a 10-point gap here as well, higher confidence in women compared to men.
Right.
And
women are more trusting.
And that is true on several other key metrics. Women do tend to, on the whole, have higher confidence in media, in corporations, which is an interesting metric, as well as in Kentucky. And Kentucky actually is very high here in their trust in public schools. I think it's above the 90 percentile in that range. So I think, and then you having been formerly in the media, I think this is a great statistic to really kind of engage here. Again, this report really tries to sort of steer away from the policy conversation, but it presents the data. And then we hope that community leaders, decision makers, political leaders, young activists, we hope anyone and everyone takes this data and then says, okay, well, what does this mean? Why is this? And then is able to go out into their community and say, what can we do to change? I think, kind of speaking a little bit more holistically, there are some other measures regarding trust in particular. Kentucky is very low. Surprisingly, on our social connectedness, or maybe not surprisingly, but on social connectedness, we have strong ties with our neighbors, strong ties with our community, but then we have low trust in our neighbors. So what does that mean? We're happy to lend a hand, but then we don't necessarily have high confidence in trusting our neighbors and community. Again, I think that may shed some light on this larger conversation of trust.
Right. Well, you have to wonder. I'm looking at the statistics now. I've got the report here. So about 46.5% of Kentuckians say they have public confidence in the media. About 63% say they have confidence in corporations, which is about the same as the figure nationally. And on public school system, Kentuckians had a 90% confidence in their public schools compared to 88% nationally, which puts Kentucky about 20th in the country. So good confidence in the public schools. But that whole thing you mentioned about not having confidence, I guess, in the neighbor and in the media, I wonder if that's the same thing, that we just don't trust our neighbor, so why would we trust the media?
Yeah, in a lot of ways, and I think this is probably indicative of more reports more generally. There's always some measures like, oh, what's the follow-up here? I wish I had additional data here. And I think that's 5 years ago, the Secretary of State's office went on a statewide campaign. They went to colleges across the state. They went to pockets of communities and sparked these conversations with the local community members. We at the McConnell Center are actually going to be hosting an event in January 26th, with more than 100 high school public students. we're trying to engage at that level. These students don't yet necessarily have the privilege of voting, but they're very active. Increased volunteerism is a key metric here in Kentucky. We definitely do like to lend a hand, but we're really, we'd like to spark a conversation there, get their thoughts. This report, unfortunately, doesn't reflect the 2016 elections, but I think I would be very curious to see confidence in media, particularly in light of the 2016
My guess is the confidence in media has gone down, but maybe volunteerism and activism has gone up, I would suspect, around the election of 2016. I'm just guessing. I don't know.
Well, I think that's definitely a great guess, and I think we're particularly interested in sparking that conversation with the younger generation, the next generation of hopefully great leaders. Again, this is a 5-year update, so we do show updates and some slight trends. There are some perhaps anomalies. Obviously, 2014 turnout here in Kentucky in terms of voter turnout was actually higher, but we have to remember. Right, it is a midterm election, so of course.
Elections. Yeah, I'm sorry. I was thinking the governor's race when only 30% showed up. I'm sorry. You're talking about the midterm elections, which was high in 2014.
But we have to recall a lot of external influence, I think, coming in from out of state money. Obviously, a very contentious race. Mm-hmm.
Yeah.
Yes.
Yeah. Okay.
So perhaps an anomaly. That's where I would say cautious interpretation, perhaps, of the data. But again, do want to highlight it is important, and it is important. I would say every 5 years. To help spark conversations and to really reflect on the data.
All right, we've only got a couple minutes left. Again, we're talking with Glippy Ryder-Jones from the McConnell Center about the 2016 Kentucky Civic Health Index. Only 11% of Kentuckians reported contacting their government officials about something, and only 23% reported talking to friends about politics. Those are really low numbers. That may have changed again this year, as we talked about with the 2016 election, but that's a really low number.
I think it's a very, arguably, very, very low number. I do think, though, that is unfortunately similar on trend to the national levels as well. But I think it may spark a conversation. I think particularly in this presidential election, we heard a lot of conversations. I'm not engaged in the political process. Again, this formal sense of engaging in politics and citizenship. We have to remember that there are both the formal mechanisms, but then there's also this informal, there are informal ways to engage as well. And I think increasingly, as we see the vitriolic and negative campaigning, and people who are disengaged with the process, that may be reflected in part of these measures, I think, as well. But I absolutely agree, very low metrics. And I think, like I said, our event in January, we're really trying to help have a conversation with this next generation. Where are they? What are they seeing? We have to remember, the 2016, particularly for the campus here at UofL, this was our very first presidential election.
Yeah,
and we, yeah.
Yeah,
So it's, this is their first glimpse. They don't have the long-range vision.
You're right. Well, I want a couple things real quickly. The recommendations out of this report: restore voting rights for felons, educate, statewide voting campaign. Those are the three things you came up with. Which of those three things do you think are the most important from your perspective to get people more engaged in politics and what's going on in our state?
I mean, I think I personally would definitely highlight this education element. Educate, educate, educate. In so many ways, I think that's what the McConnell Center's mission really embodies, particularly our civic education program, which is to offer programming, public events, public lectures that really spark conversations and really try to engage the citizenry, our campus community, our greater Louisville, and then obviously the greater Kentucky communities on what does it mean to be a citizen? What does it mean to be a citizen? There's so many lessons that we can find historically that then inform our future conversations. So really our mission is to try to connect the past to inform the present. And I think that element of educate, educate, educate reminds us that when things do seem low or negative or perhaps not as positive as they can be, we can learn how to advance and move forward by past examples. So.
All right, Nick, where I can find the 2016 Kentucky Civic Health Index report, I assume, on the McConnell Center site, and if not, just search for it on the internet, right?
Yes, we'll be launching a statewide launch come January, and trying to get this into the hands of Kentucky's political leaders, and along with our Kentucky Social Studies teacher network. So really trying to push the message and just the data to share, and again, spark the wider conversation about how do we better engage and enhance civic engagement.
Okay. Glippy Ryder-Jones from the McConnell Center, thank you very much for being on the show again. Every year, a couple million Americans get a superbug, an infection that's resistant to most drugs. But our system for quickly diagnosing those superbugs isn't very good. Ken Thompson is in the Department of Pathology and Laboratory Medicine at the University of Louisville, and he thinks he's got a test that will easily detect superbugs. Right, Ken? Is that what you got?
That's right, Mark.
Okay. Well, first of all, tell us what is a superbug? Explain what that is.
Superbugs are bacteria, and they're bacteria that cause infections. They are resistant to most or all antibiotics, and they cause mortality the same as Ebola. So they are pretty dangerous organisms. They cause infections that are different from other bacteria in that you need a combination of 2 active antibiotics to treat the infection, not just a single antibiotic. If you treat with 1 antibiotic, it's possible that the organism will develop resistance to all antibiotics, and so you've got a therapeutic failure. And in essence, the physician has 1 shot at getting the patient on the right therapy, otherwise the patient may die. So we're in the era of the septic ward, where physicians, sometimes all they can do is just watch to see if the patient gets better or dies, because there are no therapeutic options left.
And a lot of these superbugs, getting back to what a description of a superbug is, aren't a lot of these like hospital infections that are after surgery, or is it something you develop, you go to a foreign country and you get something weird? What is it?
Historically, they were mainly causing infections in hospitals and in nursing homes. But a few years ago, there was a superbug that emerged in India, and it's out in the community, and that has caused a tremendous scare because it's not just in institutions. And these organisms are actually everywhere. During the Rio de Janeiro Olympics, we learned that they were on the beaches in Rio de Janeiro. They're in the food chain, they're everywhere.
Are they getting worse across the world?
They are getting worse because we haven't been able to stop them spreading. If we could stop them spreading, well, then we could contain them.
Is that because we haven't figured out drugs that will tackle them, that these things become drug resistant?
Yes, Mark. We've had a dwindling antibiotic pipeline for many years now, and organisms have been getting more resistant. And it takes about 10 years to get a new drug from discovery through the FDA and onto the market.
Wow.
And we just don't have the treatments now that will cover all the superbugs.
I got you. We're talking with Ken Thompson, who's with the Department of Pathology and Laboratory Medicine at the University of Louisville. We're talking about superbugs, and so let's talk about what you have come up with, the way to detect superbugs. What's the problem now with trying to detect quickly when these superbugs are in someone, and then what do you think you have as a solution to that?
The problem with detection now is that most laboratories use inaccurate tests, and they take at least 18 hours for incubation, which is far too slow. Or else they may use complex, very expensive tests that most hospital laboratories choose not to use. Our test is simple, it's very easy to do, it's fast, it gives results typically in 1 to 30 minutes as opposed to 18 hours or more, and it's cheap.
And so what is this test? How can you make this test? Is it a blood test? Is it a saliva test? It's
a test that's done on the bacteria themselves. The worst of the superbugs, the ones that require 2 antibiotics, have an enzyme that is characteristic. It's called a carbapenemase. And we have a test that can detect this enzyme.
Through what? Is it a blood test, or how do you do it? If I walk in or you come into my hospital room and I'm really sick, what kind of test do you apply to me to get to know that I have the superbug?
The first thing you need is the actual bacterial isolate, so it has to be cultured first of all, and we work on the bacteria, and we detect its ability to inactivate carbapenem antibiotics, and this is the key thing to detecting these organisms.
Why is it important to detect that early?
If you detect the superbug, that it's a superbug early, then the physician has a chance to get the patient on combination therapy, and also your chances of preventing transmission to other patients are greatly increased. The longer you take to detect these organisms, the greater the chance they have to spread. So the solution to the problem is rapid detection and prevention of spread.
Okay. And in, I think the United States, was it 22,000 people or something like that die of superbugs each year?
That's about right, Mark, yes.
I think that's right. I think I looked that up, and it's 22,000 people die of superbugs. Are most of those people dying because they aren't detected early and they just go too long without getting the superbug detected in their system, or is it just the fact that it doesn't matter when they get tested, it's that these drugs these superbugs are resistant to drugs?
It's both, Mark. A lot of labs use poor tests. Some labs don't even test. And you've got to get the patients onto combination therapy really quickly.
Interesting. We're talking with Ken Thompson from the University of Louisville. We're talking about superbugs and his detection. Well, it's not a device, it's detection method for detecting these superbugs. So where at the University of Louisville did you come up with this? How did UofL fit into this? I know you work at UofL, but how did UofL come up?
How I came up with it, I married a wonderful Kentucky woman, and she came up with the idea.
So give her credit, right?
Yeah, I worked in this area for a long time, and I suggested how that she might look at devising a test, but she ignored me completely. And she's got very good instincts, and she came up with a really good test.
Okay, and she works where? What does she do?
She's at the University of Louisville Hospital, and she is a researcher at the University of Louisville in the same department as me.
We ought to give credit where credit is due. What's your wife's name?
Gina.
Okay, very good. Gina Thompson, right?
Yes.
All right. And where are you in the process here? You've come up with this test that you think will work. Has it been used on humans yet? And have you done some clinical trials? And how long might it take to get to the market?
We've done some initial tests. We have access to a collection of very well characterized bacteria, and we have used those bacteria to show that the test works. And the bacteria we've tested include some that currently used tests cannot detect this carbapenemase enzyme in. So based on studies with a small, very focused collection of organisms, we know it is accurate. We now need to do bigger studies to get the data out there and published so that we can convince a manufacturer to come along and license our test and manufacture it and sell it. We've applied for a patent. We've got a provisional patent application in. So we know from our in-house studies that it can outperform other tests, but we need to do a big study.
And when you're talking about a big study, it's a clinical trial, I assume, right? Basically, isn't it?
It's an in vitro study. It won't involve patients.
Okay.
It's to
Since you're testing
demonstrate
bacteria.
accuracy, and then we want to do a comparison against current tests to show that ours will outcompete them.
So how long realistically, before this thing is on the market? I mean, if you got to go through these tests, you got to find a company to take it on and invest in it. Realistically, how long before folks who have a potential superbug might be using this to have that detected in their cells?
I would think the earliest would be a couple of years. The University of Louisville is just preparing to go out and contact companies. We think we've got another 2 years of research to do, but based on what we've got, the university is going to see if companies are ready, want to get on board right now.
All right, Ken Thompson from the University of Louisville. Appreciate it. Keep up the good work on this project. Sounds like a good one, okay?
Thanks, Mark.
Thanks for listening, and go Cards.